Hello and welcome. My name is Holly Garioch, and I serve as the Chief Cohort Development Officer for the All of Us Research Program. Before we get started, I wanted to share some housekeeping. Closed captions are available throughout the day. To turn on captions, click on the CC icon on the top right of your screen and choose your preferred language. If you run into any technical issues or need additional accommodation, please email allabusscienceday@pyxispartners.co. To ask a question, select the Q&A option at the top right of your screen. Type your question and then submit it. Questions submitted through the chat box will not be monitored. Please be sure to use the Q&A box. Our research panel today, panel 5, will focus on brain health, mental well-being, and recovery. I am delighted to introduce our panelists. Please join me in welcoming Dr. Fatima Choupani. She is an early career researcher at Seattle University. Dr. Nari Yu, an early career researcher at the University of Michigan. And Lung Fu, a graduate student at SUNY Downstate Health Sciences University. During the presentation, if you have any questions, a reminder to please put those questions in the Q&A panel, and we'll get to them at the Q&A portion of the session at the end. We will begin today with Dr. Fatima Choupani. Thank you. Fatima, go ahead. The stage is yours.
Yeah. Good morning or good afternoon to today I present how we use graph neural network and explainable AI to uncover modifiable inflammatory drivers in Alzheimer's disease. So first, this Alzheimer's disease is driven by multiple interacting factors like amyloid deposition, tau protein, and especially chronic neuroinflammation. If you can see in the right side, is the astrocyte Alzheimer's disease. We know that astrocyte is a protective, supportive of the brain in healthy individuals, but in release of those neuroinflammatory biomarkers like interleukin 6, interleukin 5, tumor necrosis factors, it cause while losing important supportive function of the brain, astrocyte become reactive. And like glutamic uptake as well as mitochondrial damage in the brain. So what we do today, we analyzed 1462 diverse AD cases, Alzheimer's disease cases from all of us using graph neural network with APOE epsilon 4 the genomic genes. And what we found that tau emerged as a dominant, while interleukin first beta and APOE epsilon 4 stood as strong drivers. The model performed equitably, and this moves us toward personalized, modifiable interventions rather than age alone. So today we are covered the background or pipeline, key findings from traditional and AI methods, equity analysis, and transitional implications. So the background and disparities. So Alzheimer's is driven by, as we already said, multiple interacting mechanisms like amyloid, tau, especially chronic condition. The heat map in the right section, it shows that the Alzheimer's disease prevalences that among all of the people, all of us participants age 39 and higher, broken by age, sex, and race. The color intensity of the prevalence, darker means higher prevalence, and of those people diagnosed with AD. And if you can see, for example, Hispanic males age 80 to 89 has approximately 20% prevalence compared to white 2 to 3%. So these patterns highly highlight why equitable multimodal AI modeling with all of us data is so important. And our pipeline starts with multimodal data and moves toward the clustering, build a patient similarity graph, applies graph neural networks, and uses explainable AI to interpret results. So cohort selection and multimodal data from all of those participants, we identified 1462 Alzheimer's disease using ICD-10 codes. This final code is racially ethnic diverse, and approximately 27% of carrying the genes APOE epsilon 4 has the genotype, and 61% black, 61% white, 24% black, and 16% Hispanic. So the first finding is traditional statistics show age is the strongest factor, but with limited actionable insight, and which is no statistical significance. If you can see in this Cohen's D, only the age is significant and the other variable is not significant. This highlights the needs for AI can capture those complex interaction. So we uses the unsupervised clustering principal component analysis, and our multimodal biomarkers include APOE epsilon 4 inflammatory markers on our as well as metabolic factors and other inflammatory factors. So the scatter plot, these scatter plots reveals 4 distinct clusters, colored blue, orange, green, red, representing biologically meaningful subtype of Alzheimer's disease. The blue cluster in the bottom left tends to a group patients with lower tau and different metabolic inflammatory markers. The red cluster left side shows higher concentration of certain inflammatory or genetic signals, and green and orange spread toward the right, indicating patients with varying combination of tau, inflammation, and metabolic dysregulation. So these clusters demonstrate that Alzheimer's disease is not a single uniform diseases, and instead patients fall onto all these different subtypes. And building the cohort graph structure, the content is the nodes. We construct the patient similarity graph where nodes are the participants and ages represent biomarker similarity. This structure allows neural network to learn complex interaction. If you can see here, so the age node is one patient, and ages gray line connects patient with who have a similar biomarkers profile, like tau interleukin or tumor necrosis factor or APOE genes هستش. And in the color of interleukin, it shows that yellow higher in the left side, yellow higher inflammation, darker blue is lower inflammation. And these slides, the top drivers from the neural network and between APO epsilon 4 and expanded genetic risk. So this shows neural network permutation importance on raw spares. All of us data, we didn't do any imputation. So APOE from genomics as well as TREM2 and ABCA7 included in expanded genetic risk using we trained the neural network on the raw sparse data without any imputation. This chart key finding tau consistently among all of the charts. It emerges as a dominant driver across all analyses. So when we expand the genetic risk, include those other genes, modify tau importance significantly, like 1 times 1.34 higher in carriers in one analysis, 1.13 times higher in another. And SHOP explainability gives detailed insight. The Beesworm plot shows how each feature pushes prediction higher to lower. Tau has the strongest impact, while real APO epsilon 4 contributes consistently. This helps us understand the role of modifiable inflammatory pathways like interleukin first beta and tumor necrosis factor. So after that, we checked the equity. The model showed equitable performance across racial and ethnic groups with Corsall-Wallace p-value, all of them over 0.3. Is not significant. You can see not significant. And then we do an interaction between APOE epsilon 4 and future interaction. After confirming model equity, we examine to interact with other features as well as using SHOP interaction value. The left plot shows that SHOP dependence of APO epsilon 4 and tau. So you can see the effect of the APO epsilon 4 genes on predicted brain atrophy risk. It varies with tau levels and with some strong positive negative influence at higher tau values. And the right side plot, age and APO epsilon 4 genes show a complex nonlinear pattern. The impact of in is not uniform, or sex differences effects are modest. And conclusion, and so the final is, in summary, the neural network on raw sparse all of us data with APOE epsilon 4 gene identified tau as a dominant driver and highlighted interleukin first beta driven neuroinflammation as a key modifiable pathway, as well as these APO epsilon 4 genes significantly modifies tau importance when combined with other two genetic risks, TREM2 and ABCA7, this model. So the framework has a strong transitional potential for EHR-based risk scores and personalized recommendation, moving us from age-dominated models toward actionable, equitable prevention strategy. And I would like to acknowledge my co-authors as well as thank you. We are very grateful. Thank you to all of us participants as well as HARC. Thank you for your attention, and I'm happy to take any questions.
Thank you, Dr. Trapani. We're going to save questions for the end of the session. At this point, I'd like to introduce and bring onto stage Dr. Nari Yu, an assistant professor from the University of Michigan. Welcome, Nari.
Okay. Hello, everyone. Good afternoon or morning. My name is Nari Yu, as introduced. This work is in collaboration with Dr. Michael Park at Rutgers University School of Social Work and Garam Yang at UT Austin Department of Sociology. Today, I will be sharing findings on mental health condition prevalence and treatment engagement across Asian American ethnic subgroups using data from AOU programs. So before I get into the details, I also wanted to share our research summary. So we use survey data from AOU data over to 24,000 Asian American adults in AOU to compare self-reported mental health conditions and current treatment across 7 ethnic subgroups. What we found is that prevalence ranged from 8% to 20%, which is the huge range, depending on the subgroup, and treatment patterns did not track this prevalence. And some groups with high burden had low treatment rates, and others shows the reverse patterns with low burden with high treatment rates. And these patterns are completely invisible when Asian Americans are reported as a single group, which is very common practice in public health science, and which is still the default in most federal health data as well. So I also want to ground in the historical literature in Asian American mental health study. So in 2008, fewer than 9% of Asian Americans with a probable mental health disorder has sought specialty care. So 25 million Asian Americans span more than 50 ethnic subgroups, but most federal health data report them as a single category. Which community face the highest burden and which are least likely to receive care remains largely unknown. So a lot of Asian Americans actually think mental health and talking about data disaggregation is the minor issue. So it's related to the issue of minor feeling. In the words of Kathy Park Hong, described that model minority myth combined with disaggregated data patterns that mask variation has made it easy to assume that Asian Americans are doing fine in terms of mental health symptoms and treatment engagement. So we don't know actually which communities carry the highest burden of mental illness, and we don't know which communities are least likely to receive care to reach the population better and to address the problems with more concrete numbers in the in this current era. So I just showed that the number is pretty 20 years ago data. So why disaggregation matters? The push for disaggregated data on Asian American is not new, and actually it's the advocacy topic of a lot of Asian American and subgroup advocacy organizations. So you can see the data problem related to it. And also there is reported mental health gap. The well-known survey is National Latino and Asian American survey found the subgroup differences in psychiatric disorder prevalence, but is now nearly 20 years old. And subgroup studies included the subsamples, but used broader regional categories due to the small n's. So the well-known reason why Asian Americans are aggregated and reported is because of the small sample size and n. In reporting, it's the meaningful number to report it. So all of us data actually is trying to solve this issue. So we use all of us research program control tier data with the in the dates from May 2017 through 2023. And we have this sample size they have identified as self-identified Asian, and based on the sample size, we have used 7 subgroups: Chinese, Asian, Indian, Filipino, Japanese, Korean, Vietnamese, and Pakistani. And we are expecting to analyze more groups as we have more enrolled participants to all of us research program. And we use the measures outcome. First outcome is self-reported mental health conditions reported in personal and family health history survey. And second outcome is currently seeing a provider for the specific condition. We were thinking of using electronic health record, like reported visits, but decided not to use it because considering that a lot of Asian Americans are foreign born and they're like seeking care or reporting care even outside of the United States or outside of their states. So we thought that self-reported measure could be more meaningful than electronic health record linkage data, which we are going to also report it in limitation as well. So we analyze multivariable logistic regression, adjusting for age, gender, education, nativity, English proficiency, insurance status, and census region. And Chinese Americans, since they have the largest sample size, are used as a reference group to compare if we found the meaningful differences across group. So you can see the study sample summary here for the N. And this chart shows the adjusted prevalence across the 5 conditions: any mental illness, mood disorder, anxiety disorder, serious mental illness, and neurodevelopmental conditions. And the adjustments hold the constant across all socio-demographic characteristics, like you can see age, gender, and like these categories. And you can see that the variation is quite substantial. Japanese has like the highest prevalence rate for any mental illness and mood disorder and anxiety disorder, and holding Vietnamese populations at the lowest estimates. And like you can see the estimates are not overlapping each other in terms of the bar, which also shows the meaningful differences each other. So in summary, Japanese Americans, 19.6% of Japanese Americans reported any mental illness, and 14% of them also reported mood disorder, in contrary to Vietnamese American reporting any mental illness for 8% and mood disorder for 5.7%. So you can see the huge number of differences across groups in prevalence. And treatment engagement is also different across the groups. So the chart shows the adjustment treatment rates among those who reported a condition. And so the question here is, of those reporting mental health condition, what proportions are currently seeing a provider for that specific condition? And treatment rates generally range from about 53% to 79%, depending on the conditions and subgroup. But the critical finding is that the patterns across group does not actually mirror the prevalence patterns in terms of the order of the rates. So the key findings are Filipino Americans reported high treatment rates for AMI and mood disorder, and Asian Indian Americans high treatment rate for SMI, and Vietnamese Americans high treatment rates. However, Korean American, Japanese American, Chinese American, like East Asian groups, reported lower treatment engagement than other groups. So we also try to explain these patterns from the literature, why these groups report lower prevalence rates, maybe because of the acculturation alone does not explain the gap, limited Korean-speaking providers or cultural factors such as the spirituality or church-related factors, considering their factors. And also Japanese Americans, maybe they are more likely to be later generation in terms of immigration generation, so they could be associated with higher distress. And we also want to be transparent about the limitations. We use the self-reported data, and English proficiency and social determinant of health survey had large missing data, so it make some estimates unreliable, and all of us participants are not nationally representative, and they're highly engaged sample in terms of the health reporting. And Yeah, also like want to highlight that I'm really thankful for all the participants and also community organization who spent their time in recruiting diverse participants, which actually make this research possible to disaggregate the data across Asian American subgroup. Thank you all.
Thank you, Dr. Yu. Now I would like to welcome Long Fu, a third-year MD PhD student at SUNY Downstate. Stage is yours.
Sorry, everyone. Hi, everyone. My name is Lung Fu. I'm a student at the MD MPH program at SUNY Downstate in Brooklyn, New York, where I'm currently working in the Institutes for Genomics and Health. And I wanted to shout out the Data Science for Health Professions pathway, which helped make this research possible. Today, I'll be presenting our work on atopic dermatitis and new diagnosis of depression. I was drawn to this research because I have a strong interest in dermatology. And in the clinic, I saw that patients were struggling with atopic dermatitis, also known as eczema, as you can see on the right. This is the inside of an elbow, and it can get scaly, itchy, and bother patients from day to day visibly and while they're sleeping. Using my background in epidemiology, I wanted to investigate if there was a clear relationship between atopic dermatitis and mental health outcomes. The All of Us database provided me a unique opportunity to study this in the diverse US population. So to take a overall look at the research that we did, we selected around 9,000 patients with atopic dermatitis and compared them to a matched control from the general population. What we found was that people with atopic dermatitis were 48% more likely to develop depression compared to the matched control, and that the risk was highest in the first year. This matters because atopic dermatitis patients may benefit from depression screening. So a little bit of background on the two disorders. Atopic dermatitis is a chronic inflammatory skin disease. It's characterized by itchy and visible skin lesions that it approximately affects 10% of adults in developed countries. Major depressive disorder, on the other hand, is a psychiatric disorder characterized by low mood and loss of interest for at least 2 weeks, among other symptoms such as sleep disturbance, energy loss, and decreased feelings of self-worth. This can significantly impact daily life and is a leading cause of disability worldwide, with an estimated lifetime prevalence of 5 to 17%. It is thought that chronic inflammatory diseases such as atopic dermatitis may increase depression risk through shared inflammatory pathways, meaning that the inflammatory markers from the skin affects the brain's behavior, or that the chronic symptom burden, the itch, the sleep disturbance, and the visual appearance of the atopic dermatitis may affect a patient's quality of life, causing depression. Some background in previous studies. One UK study found a severity-dependent association between atopic dermatitis and new depression cases. However, this was done in a European population, and I wanted to study this in a diverse US population. So to quickly talk about how this study was created, I used All of Us controlled dataset version 8, and there were around 600,000 patients with electronic health record data ages from 18 to 65, and we excluded all patients with a history of bipolar disorder or schizophrenia spectrum disorder because these can be misdiagnosed as depression, and they're mutually exclusive with a diagnosis of depression. This leaves us with an atopic dermatitis cohort of 9,235, which we matched 1 to 4 through a process called propensity score matching, meaning we assign each person a score based on their age, sex, race, income, asthma, and allergic rhinitis. This gives us the ability to make a very similar comparison between the two cohorts. And we adjusted for asthma and allergic rhinitis because they're strongly associated with atopic dermatitis and also have their own risk for depression. So we looked at after that, we excluded depression before atopic dermatitis diagnosis. So to look forward to see if new cases of depression are developed, we had to exclude any pre-existing depression. So we were left with the baseline group of 9,000 and the forward-looking group of 7,000. But first, we wanted to look at the patients at baseline to understand where patients are starting. So let me orient you to the figure on the right. We are looking at men and women. We found some differences at baseline in depression prevalence. So the percentage is the amount of patients that have depression. The orange is the control group, matched controls, and the red group is the atopic dermatitis. So a patient comes into the clinic and gets diagnosed with atopic dermatitis. They're twice as likely to have a pre-existing diagnosis of depression compared to their matched controls, which you can approximately see from the graph. Women have an absolutely higher rate of pre-existing depression compared to men, and this is consistent with literature and what we expect. However, the one thing I would like to point out is that we found a significant finding that men with atopic dermatitis were 2.25 times more likely to have pre-existing depression compared to 1.84 times in women with atopic dermatitis. This is just a larger relative change, meaning that men are a little bit more sensitive to developing depression if they have a diagnosis of atopic dermatitis. So now to look at the forward-looking group, to draw your attention to the graph on the right, this is a cumulative incidence graph where we're looking at atopic dermatitis in red and control in blue. As you can see, and the cumulative incidence is of new depression cases over time. So looking overall at the overall picture, we see that atopic dermatitis is associated with a 48% increased risk of receiving a new diagnosis of depression. That's a significant finding. And another significant finding is, if I draw your attention to the first year here on the graph, you can see that initially the graph climbs higher for the atopic dermatitis case, for the atopic dermatitis group. This is a significant difference compared to other time periods, and it's a potential window to be addressed in the clinic. However, the risk remains elevated over time, and at 10 years, patients with atopic dermatitis still have an increased risk of developing new depression compared to their matched controls. Okay, and lastly, we wanted to look at racial and ethnic subgroups and take advantage of all of us' diverse patient population. So atopic dermatitis, our main finding is that atopic dermatitis increases depression risk across all racial and ethnic groups. And to orient you to the box on the right, every row is a separate racial or ethnic group. And the red dot is the point estimate of risk of developing depression. So 1.0, if you see the dotted line on the left, means that you're the same as the matched controls, whereas if you're to the right, the further right you are, the more risk you have compared to the control group. And as you can see, the Asian population has the largest point estimate of risk at around 2.25, where the other groups are hovering around 1.5. However, if you look at the confidence intervals, which are delineated in black, you can see that it's largest for the Asian group because it has the smallest population. But overall, the finding is that atopic dermatitis increases depression risk across all groups. And we didn't find a difference between men and women in this analysis. So looking at impact, we wanted to see, so what does this mean going forward? So physicians should consider screening patients with atopic dermatitis early for depression. Atopic dermatitis patients should be screened even if they're a long existing patient in a practice, because the risk remains high after 10 years. There is no subgroup or population that is immune from this risk or relationship to depression. And some further interesting points that we would like to study going forward if you look on the right-hand side here, we want to investigate, is depression linked with atopic dermatitis in the other direction? So does depression cause new cases of atopic dermatitis to develop? We also want to investigate the signal that we've got from the Asian population. And adjust for more variables to further clarify the relationship between atopic dermatitis and depression in the Asian population. Finally, we would like to look at how symptomology and medications affect depression risk in patients with atopic dermatitis. And that wraps it up for me. I would like to thank all the participants that made this research possible. And here are my acknowledgements, and I would like to also thank my co-authors for their assistance on this paper. Thank you, everyone.
Thank you, Lung, and to all of the presenters. Now we'd like to bring all of our speakers back onto the stage for the Q&A portion of the session. Let's see everybody come back. We have about 12 minutes now for the remainder of our time together. We'd like to take your questions and answers. If you have a question at this time for any of the presenters, please add them into the question section in the Q&A panel. The first question I see here is for Dr. Chopani. Dr. Chopani, do you have any data on Alzheimer's disease incidence in Asian Indians?
So, great question. Thank you. Unfortunately, because it's kind of racial ethnicity, usually race in all of us is self-reported, and we have an American Asian or Asian. We don't have a special Asian Indian. But a small portion, that's why I didn't include, but the prevalence of the Alzheimer's disease in the heat map shows between age of 80 to 89, 1.48% compared to black population, as well as female 1.29%. So the prevalence very low, and between 1462 participants, only we have a very small percent of the Asian. That's why I didn't include. Yeah. Thank you.
Thank you. The next question is for Dr. Yu. So Nari, were native-born Asian Americans separated from immigrant Asian Americans in the data?
Yeah, thanks so much for the good question. For sure, it's important factor in Asian American mental health research. So we didn't find enough sample size to disaggregate it by nativity, but we decided to use nativity as the covariate. So for all of the prevalence rates I have shared in the manuscript as well, nativity is controlled. So we were not able to run the disaggregated analysis, but we in the separate paper I did disaggregate it by nativity, but aggregated Asian American samples. In Asian American mental health research, everything is actually about the sample size, since we don't have enough sample size for disaggregated one. I hope it answered the question.
Thank you. There's another question for you, Nari, around statistical significance. So for the treatment differences that you saw, were they statistically significant?
So yeah, as you can maybe also see in the chart, the confidence intervals do overlap a lot with across groups. So which means that it didn't reach the statistical significance. For example, Korean Americans mood disorder treatment rate versus Chinese reference groups it was not statistically significantly different. But for some groups, we found some differences, but in general, treatment rates are not that significantly different across groups compared to the prevalence rate.
Excellent, thank you. Lung, there's lots of questions around correlation or causation from your presentation. So what are your thoughts on the reverse or reciprocal causation between AD and depression?
So running a retrospective cohort study with a population level study like this, I don't think it's possible to determine causation definitively. But the reason why we studied this direction, going from atopic dermatitis having a risk of developing further depression, is that we believe that atopic dermatitis may be undiagnosed for longer. So someone with a diagnosis of depression may carry atopic dermatitis without having it diagnosed. But yes, so what we found was there was a significant risk of developing depression, but it is not possible to determine the cause.
Got it. Thank you. And then another question for you, Long, around potential environmental exposures, around like air pollution or chemical exposures. Do you think that those could be linked to depression through the same mechanism or a different mechanism from the one that you have found?
Well, I think that is certainly an interesting idea. And environmental exposures are a big risk factor for developing atopic dermatitis. I'm not currently aware if environmental exposures can cause depression, although I wouldn't be surprised if noise pollution and some urban environments would cause a risk of increased depression. But that is something that we did not study we did not adjust for environmental factors in our current model.
And final one for you, given you're studying skin conditions, are you aware of any other skin condition association studies with depression that you might want to share with the audience?
Yes, there's many studies that are being investigated for the relationship between skin diseases and depression. There's some autoimmune conditions such as psoriasis, and certain autoimmune conditions that cause alopecia or balding. And currently I'm studying if keloids can also cause depression and whether certain treatments for keloids lead to an increased or decreased risk of depression.
Thank you. For all of the panel members, did you compare any of your other research questions to larger cohorts? Or did you just use the All of Us research program? And can you share anything around the findings that you may have found?
I believe that I compared my findings to the UK Biobank for the UK study, and I think the results were similar, although I'm not aware off the top of my head the exact numbers.
I would say like my research topic is like Asian American specific, so I didn't do it for other other countries. And there are like other data sources. They actually have like disaggregated data for Asian American, like California Health Interview Survey. But it's not, like, to my knowledge, All of Us research program is the largest data source with disaggregated data for Asian Americans. And specifically disaggregated by the symptoms and condition as well.
Thank you. It's one of the values of the All of Us research program data set that's available to researchers. Dr. Trapani, can you share a little bit about what motivated you to conduct your research?
Okay, so for the Alzheimer's disease, what is kind of, I started teaching promotion of health in older adults, and I see that Alzheimer's disease, especially with older adults aging, population this age from 65 and older, it's projecting to 1.5 billion by 2050, and Alzheimer's disease increases as well. So because one of my specialties is lifestyle and physical activity, so as well as genomic, I wanted to see how we can use this new technique, AI, and especially deep learning, to by using this All of Us big data to kind of finally build a precision medicine, individualized prevention of the disease, especially from the progression through inflammatory or changing lifestyle. So that's why one of the reasons motivated me, and my students also, because they are very interested. So that's why since 2 years ago, I started to work on Alzheimer's disease. And as well as, all of us data is very large. They have a kind of variety of variables. And I haven't compared with what I'm planning for validation to use some Ocean data to see especially some of the other variables in the future. But all of us, I think, have anything. Thank you. Thank you for the question.
Of course. Well, I want to say thank you to all of our panelists for sharing their work, spending time with us, and really demonstrating to the entire audience the powerful potential for using the All of Us Research Program data set. Thank you all in the audience for joining us. There will be a short survey following this session. After completing the survey, you'll be automatically redirected back to the agenda. Up next are the interactive workshops. There are 6 concurrent workshops to choose from. They'll all be going from 3 to 3:45 Eastern Time. The upcoming sessions are limited to the first 100 attendees. So if you're unable to join your preferred workshop, the Voices of All of Us has no attendance limit. All are welcome there, and all sessions will be recorded and you can go back to them later. So thank you very much to all the panelists and all of the audience members. Enjoy the rest of Science Day. Thank you.